As many of you probably remember, exactly 1 year ago, we collected 104 samples here at the SGI Day from approximately 30 patients, their healthy siblings, and some of the parents. Using those samples, we separated DNA, extracted DNA for genetic studies. We separated serum, which is a liquid part of the blood, for various proteomic studies. And we also separated immune cells and placed them in a freezing media, and it's a media in which cells preserve their viability when frozen. Earlier, I mentioned all these challenges that we are dealing with in our field. That includes the fact that a substantial proportion of patients with SGI do not respond to existing medications, and we certainly need new treatment targets. I mentioned that we still have a large number of patients with macrophage activation syndrome. And again, the major issue for us is the increasing number of patients with this potentially life-threatening lung disease. So we try to use those samples that we collected 1 year ago to address all the three challenges. And very briefly, I would like to list the projects that the samples were used for. So I will start with the new treatments. Certainly we use many of those samples to further understand the role of interferon gamma in the development of macrophage activation syndrome. And as you probably know, our site was opened just a few weeks ago for the enrollment of patients in the clinical trial of Gamifant. And you probably know that the first patient received this treatment here today about a few hours ago. I'm actually very nervous. I didn't sleep very well last night because we all are very anxious to know the results of this treatment. Another interesting area is IL-18. We had a long discussion about this cytokine this morning. Certainly we do believe that it has pro-inflammatory properties. There is a drug that blocks the activity of IL-18. It's called IL-18 binding protein. Fabrizio mentioned earlier the small clinical trial in adult onset Still's disease that did show some positive results. Certainly the patients with systemic juvenile idiopathic arthritis have extremely high levels of IL-18. These levels seem to go even higher when these patients develop MAS or this particular lung disease. So again, the question is, should we target this cytokine in these patients? And again, in collaboration with Scott Canna in Pittsburgh and Dr. Rafael Golbachmansky at the NIH, in fact, we use these samples to understand and create some preclinical data that would justify a trial like that. Another very interesting and very controversial area in our field is mesenchymal stem cell therapy in systemic juvenile idiopathic arthritis. As you probably know, mesenchymal stem cells are self-renewing multipotent cells that actually can differentiate, can become into various cells when the need arises. It could be injury, could be aging, but these mesenchymal cells could become muscle cells, they could become osteoblasts, and osteoblasts are very important for bone health. These cells also can become chondrocytes, and chondrocytes are cells that build your cartilage. So potentially these cells could be used for lots of different diseases, but most relevant to us is the fact that we are now learning that mesenchymal stem cells can also regulate inflammation and suppress excessive activity of immune cells. As a result, the immune cells, these mesenchymal stem cells are now being used in various clinical trials in autoimmune diseases, including rheumatoid arthritis. So these cells are easy to harvest. We can get them from bone marrow, from peripheral blood, from umbilical cord blood. They are very easy to expand in culture, and then we can infuse them back in a patient with a hope for improving the course of the disease. A couple of years ago, Nora Singer, a pediatric rheumatologist in Cleveland Clinic, secured substantial amount of money to perform a clinical trial of mesenchymal stem cells in patients with systemic juvenile idiopathic arthritis. So the money is there. However, when she applied for permission from the FDA to proceed with the trial, the permission was denied, and the FDA requested that that she produced more data showing that this specific treatment is likely to be effective specifically in systemic juvenile idiopathic arthritis. So she reached us for help, and we decided again to take advantage of the blood cells collected here in 2018 at the SGIA Day, as well as the microarray technology that allows for simultaneous measurement of activity of more than 55,000 genes that is available here in Cincinnati. And I would like to explain this technology a little bit better. So I think you all heard quite a bit about genetic studies. So in this example, we are looking at a gene. And when we perform genetic studies, we are looking for specific changes or mutations in the gene that could explain the pathophysiology of the disease. Unfortunately, in systemic juvenile idiopathic arthritis, so far we have not been able to identify really relevant variants. We found a few interesting variants that would explain some components of the disease in some patients, but in general, I have to say that the results have been disappointing. So instead of looking at the gene itself, an alternative approach would be to look at the activity of various genes at the sites in the tissues that are inflamed in the disease. And I would like to explain that a little bit better. So when a particular gene gets activated, the first thing that happens, we start seeing the synthesis of molecules called messenger RNA molecules. And then these messenger RNA molecules, they eventually get translated into a protein. So if you want to measure activity of a particular gene, all you need to do is to quantitate the number of those messenger RNA molecules. And then when the microarray technology becomes very handy. So in this example, we're looking at a chip that we are actually using here in Cincinnati. So this chip is covered with tiny little probes specific for a particular gene. And these tiny little probes have the ability to capture those messenger RNA molecules. And then there is a special screening technique that would allow quantitate those messenger RNA molecules. So using a very small sample of blood and using this particular technology, we can simultaneously assess the activity of more than 55,000 genes. So several years ago, we applied this technology and looked at the activity of gene in the peripheral blood of patients with systemic juvenile idiopathic arthritis. So just to explain kind of what kind of data we generate here. Here we are looking, the columns here are either healthy controls of patients. The patients with systemic JI are labeled blue, controls are labeled yellow. The lines are genes. Yellow means the genes is not very active. Blue means the gene is suppressed. And red means the gene is very highly activated. So we kind of generated this signature, the signature of this disease in peripheral blood samples. So what we're trying to do right now, we're trying to assess the potential of mesenchymal stem cells for treatment of juvenile idiopathic arthritis. So what we do in this particular experiment, what we did actually in this experiment, we took your peripheral blood cells that we collected 1 year ago. Remember, we placed them in that special media, freezing media. They preserve their viability, so we still can, they're still alive. So we put them in culture, and then we split that culture in two. And half of these cells are co-incubated with those mesenchymal cells. And then over time, we again repeat this microarray gene expression analysis. And the question that we're asking is, does incubation of peripheral blood cells from patients with systemic juvenile idiopathic arthritis with the mesenchymal cells brings that abnormal gene activity back to normal? So we performed several of these experiments. The preliminary results look very interesting. Not as straightforward as we expected, but again, look very promising. So we are hoping to collect more samples today, and we will repeat these experiments again with new samples, and we hope that this kind of data will convince the FDA that we can go ahead with the clinical trial. So then sort of the next area that we are focusing our efforts right now is certainly the lung disease. And In this area, we shared your samples with several groups around the country. We shared some of the samples with Dr. Carl Allen in Texas, who is a hematologist oncologist, and he's interested in the lung disease that we see in patients with other histiocytic disorders other than macrophage activation syndrome, specifically conditions like Langerhans cell histiocytosis. And he performed cytokine profiling in those patients. And again, we're trying to understand what parts of the disease are overlapping and what's different. And we think that potentially can also provide some additional clues to what kind of treatment we could use in the lung disease. We shared these samples with Dr. Elizabeth Mellons at Stanford. She's actually studying epigenetic changes in T cells and monocytes, specifically in patients with this lung disease. Dr. Danziger mentioned this, this idea of the fact that when lungs get damaged and exposed to the immune system starts producing autoantibodies against those cryptic antigens in the lungs. We don't think that it's relevant to the disease itself, but we think it could be actually an interesting biomarker. It could be used for the early diagnosis of the lung disease in patients with systemic juvenile idiopathic arthritis. And to advance this project, we are sharing your samples with a group in Phoenix that specifically is interested in this project. Another very interesting and I think very exciting project that we used your samples for is the one that is currently run by Dr. Scott Canna at Children's Hospital of Pittsburgh. And he is performing serum proteomics, generating serum proteomics profiles with the main goal to come up with a set of biomarkers that again would allow us the early diagnosis of the lung disease in patients with systemic juvenile idiopathic arthritis. Again, coming back to this picture that I showed you earlier. So we looked previously at the messenger RNA molecules. Now we're looking at the proteins. So we're measuring. So he used a specific platform called SomaLogic, and this particular platform can measure approximately 1,300 various proteins. So we assessed that in blood samples from healthy controls, patients with systemic JIA, MAS, and lung disease, patients with active MAS, patients with active SGIA and no MAS, and patients with inactive SGIA. And we also used patients with other diseases where we may see somewhat similar lung involvement, and that includes NOMID-SAVI and pulmonary alveolar proteinosis. And I will show you just a couple of examples of what kind of data these studies can produce. So in this example, we are looking at a molecule called LDH-H1, and what is interesting, so what we are looking here, patients who have systemic juvenile idiopathic arthritis and the lung disease, but no MAS. And if you look at the levels of these particular molecules in this patient population, they are much, much higher than in the other patients group. The only problem is that patients with MAS also have very high level of this particular molecule. So this particular biomarker would be good to diagnose both the lung disease and MAS, but it does not distinguish two conditions very well. So what we are looking for is something like this. Here we're looking at the molecule called MCP1. As you can see, it's highly elevated in patients in the majority of patients with the lung disease, but we don't see that much elevation in other patient groups. So that could be a very promising biomarker for the early diagnosis of the lung disease in patients with systemic JIA. So this project actually produced quite a few molecules like that. So we're hoping that very soon we will be able to come up with a diagnostic chip, if you wish, that would use this kind of biomarkers to distinguish patients with emerging lung disease as early as possible. So Grant gave a long lecture today about the lung disease studies that we conduct here at Children's Hospital Medical Center. And if you remember, the central part of his talk was this gene expression analysis in inflamed lung tissue. So in this particular experiment, he used the entire piece of the lung tissue that is composed of lots of different cells. And that type of analysis did implicate interferon gamma in the development of lung disease. So now we need to understand a little bit better where this interferon gamma is coming from, what type of cells are responding to interferon gamma. So we think that the next step here might be another technology that we call single-cell RNA-seq. So this technology allows assessment of more than 55,000 genes, not just in a whole tissue sample, but in each separate cell. So in this particular experiment, Grant separated approximately 1,000 cells from the leftovers of a lung biopsy from one of you, and then he generated gene expression profiles for each of those cells. So imagine the amount of data that we are dealing here with 1,000 cells, and we have approximately 55,000 genes assessed in each of those cells. So he looked initially at this data, and he, in fact, in this experiment, was able to distinguish the population of macrophages, which consists at least of 3 different subpopulations. He distinguished T cells, B cells, he distinguished lung cells. Some of these lung cells are clearly producing surfactant. He also could pick up some cells that line the epithelial cells in the lungs, and cells lining the blood vessels, and there are some other cells that we are still trying to understand exactly what they are. So now the next step is to focus on this cell population and analyze those gene expression profiles separately, and we hope that this kind of analysis will help us understand what type of genes get activated in the inflamed tissues, and that potentially could lead to some new ideas about how the disease could be treated. So we really hope that when you are here next year, we might be able to propose some new ideas about the treatment of this disease. My point is that none of these studies would be possible without your participation. And I'm particularly thankful, by the way, to the siblings. It's very difficult to get good, healthy control samples to study disease. And siblings, it's an ideal situation. These kids they live in a similar environment. They probably have a very similar genetic background. The only difference is the absence or the presence of the disease. So for us, that collection of healthy control samples from the siblings is very, very precious. And I understand that for lots of them, it was a big sacrifice. But again, I would like to stress how important it is for us to be successful with this study. So thank you very much for your attention.